Immunoprecipitation is a very powerful technique to isolate and purify a target protein. In smooth conditions, partners, regulators, or substrates may be coimmunoprecipitated. A new interaction network may then be discovered. The activity of the immunoprecipitated protein may also be assessed. In particular, activity of the kinase may be tested on different substrates by p32 ATP labeling.
Nevertheless, activity of inhibitors targeting a kinase involved in a disease may be evaluated. They may constitute lead compounds to develop new drugs. This method can be extended from mammals to yeast or bacteria. This technique is not that difficult. 2 key points. Ensure to have a negative control to be sure the detected interaction is specific, and carefully choose lysis conditions to keep interactions and activity. Small details and gestures are important to ensure the success of this technique, and they are never described in material and methods of articles.
Demonstrating the procedure will be Fabienne Godin, a technician from our laboratory.
After cells are prepared in the cell culture room within the biosafety cabinet, use a 10 ml pipette to remove the media from the plates and discard it in a dedicated waste bottle with bleach. Then add 10 ml of fresh supplemented DMEM and put the plates back into an incubator at 37 degrees Celsius. To prepare the transfection mixture, in a 15 ml tube, add 450 microliters of